Hi, my name is Røgen and welcome to my virtual poster presentation. If you'd like to try the software yourself, you can go to the server following this link. In version 0.5 there are links to YouTube videos explaining how to operate the server and explaining the theory behind, which I'll mostly skip now. The program takes an alignment and superposition given by another program of protein structures. That program has then scored the distances that all the alpha carbons travel when doing the linear interpolation between the one structure and the other structure. So the program checks if we are following that morph, if there are going to be steric clashes, and if the backbone of the protein is going to pass through itself. Now, the one way, a typical way of this happening is if you had a right-handed loop in one structure and a left-handed loop in the other structure. The ISMB superposition, for instance, would suggest that you should just pass through yourself here. That's the shortest path from the one structure to the other one inside R3. But in the configuration space, you actually need to take a detour and in order to get from the one configuration to the other one. So this is what the program finds, if it's possible, if there's room for doing that, and it gives you an estimate on how long a self-avoiding morph is from the one structure to the other one. The self-intersections that it cannot resolve are called essential, and being essential in the sense that it involves rearranging more residues than you as usual allow the program to use. And you could, for instance, consider here two homological cath domains. There's a self-intersection in the morph between them here. And in order to get from the one configuration to the other one, you need to rearrange the 60-70 residues. Here's a big part of the protein, and the program can find out you can do that, but probably you wouldn't allow it to rearrange that many amino acids. Well, are protein folk classes generally separated by such topological barriers from morphing one structure into the other one? To answer that question, I'm taking relatively short cath domains clustered at 60% of the sequence identity, and the entire smaller domain is morphed into the bigger domain, but the bigger domain is only allowed to be up to 10, have more than 10%, to have 10% more residues than the smaller one. So the domain should be fairly identical if classified as being homologous or a chain topology class. The bigger rearrangements you allow the program to use, the more the self-intersections it finds can be resolved by finding a detour inside the configuration space. If you allow in the order of 15 to 20 amino acids to take place in these rearrangements, you would find that 10 to 15% of these homology classes are actually separated by topological obstructions where you need to rearrange bigger parts of the protein. For topology classes, we are talking about 30 to 40% of the cases where there's no simple morph from the one structure to the other one. On the other side, most morphs are between topology classes because that's the most cases you find. They are about 40 to 30% of the time you can actually find a self-avoiding morph quite easily between the structures. So this emphasizes the continuous nature of protein fold space. Couldn't I just use a normal alignment score to tell me that there are such topological obstructions? Well, when I submitted this first, TM-Align was the most restrictive aligner. That was actually in 2015. It only aligns residues within 5 angstrom in optimal superposition. In the 140,000 alignments done here, there are 40,000 self-intersections between the aligned part of the structure. So even with this restriction on which amino acids you align, there are going to be topological changes taking place in the aligned parts. Also, you can see here I can construct configurations for very small deviations in structure, where you change the threading or could even change the fold class completely. So theoretically, there's a very short distance to configurations with alternate threading and even knotting, if you like. So if you're interested in protein model refinement, you might find this interesting, because you can see here more than half of all the models submitted to the last CASP are actually separated from the target by self-intersections in the morph. And whenever you have those, you cannot take the direct route down to the target structure, but you actually need to take a detour that usually would send you out on a larger RMSD or a lower DDTS score in order to refine the structure and get down to the native structure. So this means that the most models are actually not lying in a basing of attraction when seen in a DDTS score, for instance. We offer an estimate of how long the path is inside the configuration space, which actually I think better tells you how far away the model is from the target. The ongoing thing is instead to focus, well, how big alignments can I get such that it's topologically possible to get from the one structure to the other one. So it's optimal topological obstruction free alignments. That's going to be the next output. Hope to see you in